Friends, welcome to my YouTube channel Journey with VIJAYKUMAR SHRIVASTAV. Today we are going to present the topic Principles of Hybrid Seed Production. First I would like to introduce myself. I am VIJAYKUMAR SHRIVASTAV. I have done my M.Sc from Pant University of Agriculture and Technology with a specialization in Agronomy in 1996, and presently I am working as a seed professional in one of the M.Sc which deals with seeds and pesticides. In this presentation, we have covered the various factors and topics which are playing very important role for maintaining genetic purity of seeds as well as physical purity and maximizing seed yields and crop yields. This presentation is very helpful to the candidates who are preparing for agriculture competitive exams, general competitions, and worth of knowledge for agriculture professionals and seed professionals. I would like to get feedback from all of you for further improvements in my next coming presentation. Now let's move for the presentation.
Seed production. Production of genetically pure and otherwise good quality pedigree seed is an exacting task requiring high technical skills and comparatively heavy financial investment. During seed production, strict attention must be given to the maintenance of genetic purity and other qualities of seed. Seed production must be carried out under standardized and well organized conditions. Importance of seed production. Multiplication of superior varieties, availability of superior varieties to masses, maintenance of genetic purity, supply of high quality seeds. Seeds have high germination status. Seeds having vigor and viability status. Maintaining excellent health status of seed and securing high crop yields and income level to farmers. Here we have shown the important safeguards for maintenance of genetic purity. We can describe this as control of seed source and use of approved seeds only in seed multiplications. Inspection and approval of fields prior to planting. Providing adequate isolation to prevent contamination. Seed multiplication outside their normal cultivation area. Rogging of seed fields at critical stages. Periodic testing of varieties for genetic purity. Certification of seed crops to maintain genetic purity and quality of seed. Frequent field visits and monitoring during all operations and grow out tests. Now in these slides, we have gone through terminologies used in hybrid seed production. Seed. Seed is a plant part having living embryo that have a capacity to produce identical plant and used for raising seed crops or commercial crop. Seed stand for any of the material used for sowing or planting like seed, seedling, tuber, bulbs, rhizomes, roots, cuttings, grafts, and any other propagating material for food crops, cattle fodder or floriculture. Now we'll discuss about hybrid seed. Hybrid seed, the seed F1 generation produced from a cross between genetically dissimilar parents, and the hybrid vigor or heterosis is the absolute phenomenon where expression of the characteristics such as growth related traits, flowering, and yield fall outside the range of its parents with superiority. Germination. Germination is the ability of a seed when planted under normal sowing conditions to give rise to a normal seedling and developing into new plant. Sowing. Sowing is the process of placing or planting seed to the soil so they may grow. In sowing, generally little soil is placed over the seeds, and sowing can be hand sowing or by machines. Now we have discussed here isolation. The state of being in a place or situation that is separate from others, the condition of being isolated is called isolation. And in seed production, there are three types of isolation. First one, distance isolation, to be maintained. Recommended isolation distance is per crop standards. And time isolation, to be followed to escape flowering simultaneously within seed and other plot to avoid contamination. Barrier isolation, topographical surface features, natural obstacles like tall crops, and artificial obstacles like tarpaulins or plastic seeds are used. Seed rate, the number of or quantity of seeds planted per unit of area to ensure optimum plant population for maximum yield. Spacing. Spacing refers planting or sowing of seeds, maintaining desired distance between row to row and seed to seed. Now we'll discuss about staggering. Staggering refers differential sowing of male and female parents to achieve proper synchronization of flowering of both parents during hybrid seed production. Staggering depends on flowering behavior of both male and female parents. Rogging. Rogging refers to the act of identifying and removing plants with undesirable characteristics from agriculture or seed production fields. Rogues are removed from the fields to preserve the quality of the crop being grown. Planting and sowing ratio. It refers to the sowing arrangement of row ratio of female and male lines. Like example 4:1 means 4 lines female and 1 line male. This is followed in hybrid seed production where two parents, male and female, are used. And the planting ratio or row ratio depends on different various factors like growth and behavior of pollinator, plant height of pollinator, size of male panicle and amount of residual pollen, stigma exertion of CMS female line, duration and angle of floret opening of CMS female line, and prevailing wind directions and velocity during anthesis time. Synchronization. It refers to the simultaneous flowering of male and female parents in hybrid seed production. Synchronization is flowering between the parental lines assumes greater importance as the seeds set on female parents depend on the amount of pollen supplied from the male parent during flowering period. Flowering. Flowering refers to the full blooming of the reproductive parts of the plants where pollen moves from male organ to female stigma for pollination. Anthesis. The period during which a flower is fully open and flower becomes sexually functional. Anthesis refers to a series of events between the opening and closing of spikelets. Anthesis will occur when a portion of the panicle is excited and it starts with the spikelet at the tip of the florets. Emasculation. Emasculation, removal of male organs, stamens or anthers, killing the pollen of a flower without damaging the female reproductive organ is known as emasculation. In bisexual flowers, emasculation is essential to prevent of self-pollination. In some crops, hybrid seed production like cotton, okra, chili, tomato, etc., emasculation procedure is followed to make female sterile. Pollination. The movement of pollen from the anthers to the stigma by wind is called pollination. Female stigma is essential for seed set and therefore crucial in seed production. In case of hybrid seed production, pollen transfer from male parent to female plant stigma. The movement took place through wind blowing. Now manual crossing. In case of hybrid seed production, pollen transfer from male plant anthers to female stigma manually. It is followed in hybrid seed production of the crops where emasculation is done like cotton, okra, chili, tomato, etc. Supplementary pollination. Supplementary pollination serves to enhance the outcrossing rate in order to increase seed set. Supplementary pollination should be done by shaking the plant pollen parent with the help of ropes or sticks so that the pollen is shed effectively on A-line plants. This is generally followed in hybridized seed production. Harvesting. Harvesting is the process of cutting and collecting the mature crop from the field. Picking. Picking refers to the manual collection of ripened plant produce. Post-harvest operations. Post-harvest handling is the stage of crop production immediately following harvest, including drying, cleaning, sorting, and packing. Grow-out test. Grow-out test conducted to determine genetic purity of a seed lot wherever it is prerequisite for grant of certificate, and also on the seed lots where a doubt has been arisen about the genetic purity. The grow out test can be complemented by certain related laboratory tests also. Now we'll go through the process of hybrid seed production. The first, field selection. The fertile, well developed, well labeled, with good irrigation and drainage system field should be selected, where same crop was not grown in previous season, to make free from volunteer plants, to avoid admixture and any quality issues related to genetic as well as physical purity. Field preparation. The field should be fertile and well prepared for good germination and good crop growth. Seed. The foundation seed should have quality as per seed standards, and it should be from truthful sources. In case of OP varieties, open pollinated varieties, seed production, we have to use single parent, while if we are going for hybrid seed production, we have to use two parents, female A line, which is cytoplasmic male style line, CMS line, and second, male restorer R line. In hybrid seed production of some crops like maize, pearl millet, sorghum, rice, mustard, sunflower, castor, sorghum Sudan grass, etc., female parent A line is sterile parent. While in some crops like cotton, okra, chili, tomato, etc., we are following emasculation procedure to make female sterile. Below points are important in hybrid seed production. First, CMS A line desirable characteristics. B, restorer R line male desirable characteristic, and hybrid F1 desirable characteristics. Now we will discuss here the desired characteristics of lines used in hybrid seed production. First, A line. It should have a stable and complete male sterility, good out crossing ability, good combining ability, and desirable agronomic characteristics. If we will discuss about restorer male R line, restorer play an important role both in increasing heterosis as well as in increasing hybrid seed yield seed production plots. The desirable characteristics of R line can be described as a strong and stable restoration over seasons and locations, highly vigorous plant growth habits with large number of spikelet, complete dehiscence of anther and high amount of residual pollen, profuse tillering, synchronous heading, and concentrated blooming of spikelet, multiple disease and pest resistance, good outcrossing and combining ability, and acceptable grain quality. In this slide, we have shown the prerequisites for a good hybrids. Means for producing a good hybrid, successful and good hybrids, there are certain parameters to be followed, and it is the requisites like a stable cytoplasmic genetic male sterility system, adaptability across environments, perfect synchronization in flowering, height of the male and female parents, stigma receptivity and pollen dispersal, ease in seed production, high-end economical seed yields, high level of yields on commercial scale crop. Now here we have shown the stages we are following in hybrid seed production. As earlier shown, we are using two parents, A line female parent sterile and R line male parent, and outcrossing. After outcrossing, we are getting the F1 seeds, which is used for commercial crop growing. And there are different stages. It starts from sowing, moves to vegetative, then bootleaf stage, flowering stage, cross pollination, seed development, maturity and harvest stage, then processing, cleaning, and it results to F1 seeds, which is actually hybrid seeds used for commercial growing of the crop. And in this photo, we have shown the arrangement of male line, female line. This is the photograph of hybrid millet seed production, where generally we are using 18 lines of females and 2 lines of male. And these two male lines are called R line, and these in between female lines are A line. After crossing, it will result to hybrid seed. The female is sterile. Now, strict isolation. Whenever we are going for hybrid seed production, isolation is the one of the primary requirement to get good quality seeds and avoid contamination. There are isolation standards varying from crop to crop, and for best genetic purity, it should be complied strictly. Sowing method and spacing. Standard sowing methods are followed as per crop recommendations. Depending on parents' idiotypes, we have to arrange row and plant spacings to maintain optimum plant population for high yield. Staggering. Proper staggering should be adopted based on flowering behavior of parents in producing environmental conditions for perfect synchronization. That's why there are differential sowing of male and female. We need both parents same time flowering for the best seed setting, which will result to automatically produce yield. Now planting ratio. Correct sowing arrangement ratio of female and male lines should be followed for optimum plant population of both parents based on outcrossing ability of female and pollen supply capacity of male. Female population is important for seed yield and male population for sufficient pollen supply and outcrossing. In different crops, different planting ratio followed depending on parental characteristics. Field monitoring. Scheduled field monitoring is very important for supervision of crop from sowing to harvesting to ensure timely operations for best quality seeds and good seed yield. Good seed yield. It's also useful for maintaining the historical data for the experimentation whenever needs. Agronomic management. It refers proper management of crop, supplying fertilizer, nutrition, and irrigation timely, and agronomic practices. Good agronomic management results to good yield and quality. Now we'll go for plant protection. Plant protection refers disease and insect pest management timely to prevent damage to the crop. Appropriate steps to be taken timely for good yield and quality maintenance. Rogging. It should be done timely to rog out undesired plants as off types, pollen shedders in A line, semi-sterile plants, volunteer plants, and other crop plants and disease heads, etc. For effective rogging of off types, it is necessary to have knowledge of the distinguishing characteristics of identification of A and R lines. And there are different stages on which we have to do rogging. It should be done timely and prior to any contamination. Rogging is done from vegetative stage to harvesting based on many parameters as morphological and reproductive characters. Even the critical stages are most important as below: vegetative stage, flowering stage, pre-flowering stage, and maturity stage. On these stages, compulsorily we have to do rogging. Now synchronization. Crops should be in frequent observations to check nicking, synchronization status for same time flowering. There may be little variation in flowering based on climatic condition and flowering behavior. So in such cases, corrective measures should be taken. Male lines today's advancement is recommended for good quality and seed setting. Flowering. It refers to the full blooming of male and female floral sexual parts and pollination takes place. If we are following correct staggering and there is proper synchronization, it will be results best seed setting and highest yields. Now emasculation. This practice followed in cotton, okra, tomato, chili and other crops where female line is not sterile. If female is sterile, there is no need of emasculation. Pollination. In case of pollination, there is transfer of pollen from male plant to female stigma. Good pollination is very important for good yielding. Wind velocity and wind direction plays important role in pollination. and rainy weather conditions will reduce pollination effectiveness. Generally, pollination took place with wind, but in case of crops as earlier where emasculation we are doing manual pollination, means we are using labors for transfer of pollen to the female stigma. Manual crossing, manual pollination, as earlier discussed, it is done in crops where emasculation process is done and labors are transferring pollen after collection from male parent and crossing to female stigma. Supplementary pollination for effective pollen dispersal during peak flowering stage for a specific period is fixed, like how many days and how many times in a day, for maximizing out crossing for proper seed setting. Generally, this practice followed in hybridized seed production. Seed development after crossing pollination, there will be seed formation and development. Seed maturity refers to the physiological and functional changes that occur from time of anthesis until the seeds are ready for harvest. Compulsory we have to go for harvesting after seed maturity, which will result very good quality of seed size, this is germination status, vigor, and good crop stand. And there should be optimum moisture while harvesting. If we are going for early harvest, there may be some issues related to germination and durability will be go down. So correct stage is very important for maturity and harvesting. Physiological maturity is defined as the occurrence of maximum seed dry weight and represent the end of dry weight accumulation and seed filling period. Now harvesting. In case of seed fields, first we should go for male harvesting, and after male rows inspection, female rows should be harvested, and necessary care need to be taken to avoid any admixture. Picking, followed in cotton to pick seed lint from busted balls, and in vegetable crops for separating fruits. Post harvest operations, after harvest, drying to be done to achieve optimum moisture percent, proper threshing to be done to avoid seed damage, and bagging with proper labeling. After harvesting, post harvest operations, we are moving threshing, which is the process of separation of seeds or grains from a spike or cob after seed maturity. And harvesting. Ginning. Ginning is the mechanical process of fudge seed separation from lint. This process is followed in cotton crop produce. Delinting is the process of removal of fudge from cotton seed. This is the chemical process where acids and gases are used. After that, we are going for plant processing, which refers to the mechanical cleaning to obtain maximum percentage of pure seed with maximum germination potential. And last, storage. Whatever seeds we have produced, it has gone through different crop stages and different process. Ultimately, the harvested seed will go to storage, and the seed should be stored in favorable conditions, maintaining identity, maintaining purity, protection from insect pest, maintaining germination with regularly check. In we have mentioned all steps here in seed production. So to maintain the genetic purity, it is compulsorily we have to adhere all these parameters, which will result good seed, and ultimately we can increase the storability and viability and vigor, everything, and ultimately there will be good crop stand and high yielding. About process in seed field. Now there are some key issues which are arisen in seed production, and they are related to different stages and different factors, like field related. There are issues with wrong field selection, isolation issues, poor crop management, wrong nicking, disease and insect problems. Same quality related issues like off types, pollen seeders, volunteer plants, mechanical mixtures during threshing, harvesting, or other operations, and harvest moistures. After that, people related issues like unskilled seed producers, production cost related means procurement prices, weather related factors are drought or excess and untimely rains, seed discoloration, rain damage, germination issues, disease and insect problems. And after that, there are some issues related to operations like sowing time, rogging time, harvesting stage, post harvest operations, mechanical cleaning like threshing, harvesting, drying, bagging, gunny bags, lot marking, and dispatches, etc. Ultimately, there are key deliverables of efficient seed production technology like highest order of seed quality in terms of genetic purity, seed vigor, and germination, maximization of seed yield per unit area, reduction of production cost per kg of seed, freedom from inert matter, wheat seed and other crops, seed as per prescribed standards, freedom from seed-borne diseases, and timely availability of seed to customers. These are the ultimate results of efficient seed production. So this was all things about seed production we have gone through. Now my presentation ends here. Thank you very much for your kind attention and see you again shortly. You can go through my YouTube channel Journey with VIJAYKUMAR SHRIVASTAV, where I have already published the contents like seed science and technology basics and seed development programs and seed and agriculture organizations. And this is my third one, principle of seed production. Thanking you again. See you.